Hello, before we begin, are there any questions? Can somebody confirm that you can hear me?
Yes.
Thank you. Let's see, let me share my screen. You should be able to see my screen now. I might not have my syllabus up here yet. Um, uh, actually, there it is there. It looks confused. I already know what we're doing, but I would like to show you. We're going to be covering chapter 1 today. We're a little bit behind schedule. Actually, we're supposed to finish chapter, no, just on chapter 1. So let's get to it if there's no questions. Usually I give you an overview, such as this slide here and the next slide, where I give the major goals of the lesson and a rough outline. So know the terms of this lesson. We're going to then talk about, actually, we'll move into the importance of microbes next. And we'll talk about this throughout the lesson, as well as we'll go into a brief history of microbiology. Any questions about what we're covering? All right. So when we're talking about the importance of microbes, obviously they are important, but you probably don't know exactly how important microorganisms are.
Can you
Here we're
record us?
looking at the nitrogen cycle, where nitrogen starts on the planet Earth and then where it moves from one area to another. Like you can say nitrogen starts in the atmosphere, is into gas, and into gas can be fixed by bacteria and archaea into organic compounds where the nitrogen is put in an amino group. You don't need to know that part, just that nitrogen is an organic compound. Most of that nitrogen will then decompose when the bacteria or archaea die, and it'll be decomposed by other bacteria, archaea, and then fungi. So mostly decomposed by microbes. And it'll be decomposed into ammonia or into ammonium. And you just need water around, and ammonia can convert into ammonium. This is a form of nitrogen that green plants can use, but they don't like it. And if you get too much ammonium or ammonia on the green plant, it'll actually die. The ammonia or ammonium can be changed into nitrite by bacteria and archaea, and then nitrites, that's NO2 with a negative, can be converted into nitrates, that's NO3 with a negative. And this is the form of nitrogen that green plants love. In fact, if you ever see fertilizer, you'll notice that one of the main ingredients of fertilizer is nitrate. And then green plants can take that form of nitrogen up as well as some ammonia, and then they'll convert it into organic compounds. And most of that will decompose into ammonia, although some of it can be decomposed into nitrates. And then nitrates, so we'll continue around this way, nitrates can be converted into nitrogen, which will then go up into the atmosphere. And once again, it's bacteria and archaea that can convert nitrates into nitrogen. So how important are microbes in the nitrogen cycle? Wherever we have a red arrow, this step of the nitrogen cycle is happening because of microorganisms. And we'll use the term microbe and microorganism synonymously, at least in this class. So you can see there are a few steps in the nitrogen cycle that do not use microbes, but most of them do use microbes, like this step and that step and this step. There is an exception, and that is the uptake of nitrate or ammonia by green plants. Doesn't require microbes. And then when green plants decompose, that's done by mostly microbes. So most of the steps in the nitrogen cycle are running because of microorganisms. And if we didn't have microorganisms in the planet Earth, nitrogen would not recycle. It would accumulate in one place or another and then just stay in that form. And so more and more nitrogen would come into that form. Any questions about the importance of microbes and the nitrogen cycle? If not, let's talk about the importance of microbes in making soil. There are actually rock-eating microbes where the microorganisms obtain nutrients from rocks. And these microorganisms actually break down rocks, leading to the formation of soils. Now, most of these microbes release an acidic compound which breaks down the rock, but you can break down the rock in other ways. This is not the main way that soil gets made, at least on the planet Earth. The main way that soil gets made is by the erosion of wind and water wearing away at the rock. But some rock can be converted into soil by microorganisms. So microbes are important in the formation of soil. Any questions about that?
I do have a question about
They're
the
also important in the atmosphere because it's because of microorganisms that we get oxygen in the Earth's atmosphere. Now, it is true the green plants do make some of the oxygen that we have in the atmosphere, but only about 50% of the oxygen that we have in the atmosphere is from green plants. The other 50%, and that's 50% or 21% of the atmosphere, is because of the working of microorganisms producing oxygen, releasing it into the atmosphere. And then if we go back in time, we'll see that the Earth's atmosphere had less and less oxygen until we come to about 2.7 billion years ago, and there was no oxygen, or essentially no oxygen in the Earth's atmosphere. And from, 4.6 billion years ago when the Earth formed to 2.7 billion years ago, there was no oxygen or essentially no oxygen in the Earth's atmosphere. And then what happened between 2.7 and 1.8 billion years ago, when oxygen first started accumulating in the Earth's atmosphere? What happened was the evolution of microorganisms that produced oxygen. And about 2.7 billion years ago, we had photosynthetic microorganisms evolve, and then they released oxygen, which then went into the atmosphere. And that started the Earth's atmosphere having oxygen. With this start, green plants could then evolve. Before that, green plants could not evolve. And in fact, before that, you couldn't have life on the surface of the Earth, at least on the land, because without oxygen, UV light would come through the atmosphere, and then that would kill life. But because of microbes, we got oxygen, and that led to the evolution of green plants, which together, green plants and microorganisms, brought the oxygen level to what it is today, which is extremely important to us because we have to live with 21% oxygen. And if we don't have that, just a little bit under 21%, then humans cannot survive. So microorganisms are extremely important in the generation of oxygen in the Earth's atmosphere. Any question about that? Microorganisms are also involved in the sewage treatment. And this is a sewage plant, which humans have obviously made. And of course, humans make the waste. All of this water comes from when you flush your toilet or you send the water down your sink. It's taken to the sewage treatment plant and then recycled. And it's the humans who recycle the wastewater, right? Anyone object? You should. Humans do not recycle whatever we flush down the toilets and wash down our sink. It's actually microorganisms that are growing in these vats, which break down the waste material in the water that flush down our toilets. Any question about that? So all humans do is we make a place for the microorganisms to break down the sewage water. Humans don't recycle the sewage water. It's the microorganisms that do that. All right, any question about any of that? The point is, we've shown you now at least 3 examples where microorganisms are important to the well-being of not only humans, but to the well-being of life on Earth. Let's talk a little bit about microorganisms, which we sometimes call microbes, meaning we use the 2 terms interchangeably. These are organisms which are too small to be seen with the unaided human eye. They are largely single-celled organisms. They take on many shapes and sizes. They include bacteria, archaea, yeast, mold, protozoans, some algae, like the microscopic algae, single-celled algae. If you're talking about something large like kelp, which washes up on the Washington and Oregon beaches, that is not a microorganism. Microorganisms also include viruses and prions, which are a little different. Viruses and prions are special because they do not have cells. We do call them a microbe. Okay, I'll try not to call them a microorganism because they really aren't an organism if they don't have cells, but people do call them microorganisms. But I'll try and call them microbes. Okay, they are special because they're not cellular. You could even argue, are they alive or are they not? We're not going to talk about that. Microorganisms are almost everywhere. So we say that we have a ubiquity of microorganisms. They play many beneficial roles in numerous processes on the planet Earth. They are also numerically the most abundant life form on the planet Earth. Any question about any of that? All right, I'm not hearing any questions, so let's move on.
I do have one question.
Huh?
Can we get a copy of this PowerPoint?
You can get a copy of the PowerPoint after the lesson is finalized. I want you to make your own notes because you guys, or humans in general, learn better from your own notes. I do not want you just to have my notes. Okay? But I will make my notes available to you after we are finished with the lesson. And you can print out the study guide. It has the same material I'm going over. It's just that material is in sentence form. It's not in PowerPoint form. All right, let's move on to talking about what a cell is. I've talked about a cell a couple of times, and so we better define it. A cell is the basic structural and functional unit of all living organisms. Now, a virus doesn't have a cell, and we mentioned that. It's something different, but we can say that a virus is not an organism. Cells are highly organized compartments bounded by a thin, flexible structure that we call the plasma membrane. All cells have three components. They have, of course, the plasma membrane, which you already talked about. They have cytoplasm, and they have DNA. Any question about any of that? All right. So when we're looking at these pictures here, where would we find microorganisms? Would we find microorganisms in the water, in the plant life or the other life, on the rocks? Nobody's
We
going to
find it everywhere.
How about in the open ocean? Would we find microorganisms there? Now we're talking about 1 cubic centimeter, so a little less than a half of an inch. Now, how about in the air? Would we find microorganisms there? And in the plant matter, the grass or whatever, the vegetables, we do have microorganisms. But once again, do we have it in the air? Do we have it in the soil? Are microorganisms found in the soil? And in this picture here, do we have microorganisms on the person and that animal on top of him? How about in the open desert? Do we have microorganisms in the sand as well as in the air? And in this picture here, that's a volcanic vent. That's red lava and then rock around it. steam coming out. Do we have microorganisms here? Anyone want to take a guess?
Yes.
You're correct. We have microorganisms everywhere except right here where we have the lava. This would be too hot for cells and for DNA to survive. So there's no microorganisms here. But we have it in the water in the plant. We have it in the ocean. We have it in the air too. Microorganisms you can find floating around in the air up to about a mile high. We can find microorganisms on plants and in the soil. And you can go down a mile into the soil before you stop finding microorganisms. Of course, we have microorganisms on people or on the cat. We have it on the sand in the desert and in the air of the desert, and then the rocks of the volcanic vent, and in the air, and everywhere except where the lava is. So microorganisms are almost everywhere on the planet Earth. Any questions about any of that?
I actually have a question. Is there a certain temperature that is too cold for microbes?
I'm having a hard time hearing you. Let me turn my volume up.
Oh, sorry.
The volume.
Is there a certain temperature that is too cold for microorganisms?
It depends on what the microorganism is.
Okay.
Some microorganisms can live above boiling water.
Okay.
And I forget the exact temperature. I think it's something like 126 degrees Celsius. Some microorganisms can survive that temperature. Oops.
All right.
Okay. And then other microorganisms like E. coli, they will die in very hot water. I'm talking about water that you get out of your faucet. So it depends really on the microorganism. And then there's some microorganisms which only live in refrigeration temperatures. So they'll be alive in your refrigerator, but then when you bring the item out and it warms up to room temperature, the microorganisms will die. So it depends on the microorganism, but the highest temperature we know of a microorganism growing, meaning staying alive and actually growing, I think is 126 degrees Celsius. Okay, you don't need to know that, but it was a question that was asked.
Thank you so
Shrink
much.
that down a little bit. There we go. So where is everywhere? Microorganisms are found on the land and in the water, in the atmosphere. And as I stated, you can find them up to a mile in the air. You can find them below the land and under the ocean. And once again, you can find them in the ground, almost a mile down in the soil. Now, if it's rock, of course, it won't go down that far, solid rock. And in the ocean, it's the ocean bottom. You can go down a mile underneath the surface of the ocean bottom, and you can still find microorganisms. You can find microorganisms on plants and animals. You can find microorganisms growing essentially anywhere where there is liquid water present. And you will find them between temperatures at a minus 20 degrees Celsius to at least 120 degrees Celsius. These organisms, if they're growing, they have an antifreeze in them. And that's why they can grow below freezing. And these organisms are just organisms that grow at high temperature. And yeah, the very highest is 126 degrees Celsius. Okay, but that's, I don't know. That's the record keeper. Any question about any of that? All right, microbes are numerically the most abundant life forms on the earth. How many microorganisms are there? When we're talking about 1 gram of soil, which is about 1 centimeter by 1 centimeter, you can find around 1 times 10 to the 8th prokaryotic microorganisms, meaning mostly bacteria and archaea. If you were to count viruses in that, it would be about 10 times higher, but we're just going to talk about cellular microbes. We'd find about 1 times 10 to the 8th cells in 1 gram of soil. And it will depend on how much organic matter is in the soil and how moist the soil is. So you can find less in very dry desert soil with very little organic matter. And then if you found, I don't know, something like a bog with decaying organic matter in it, you might find a little higher than 1 times 10 to the 8th. Now for 1 milliliter, which is about 1 gram of ocean water, would you find more or less microbes than in 1 gram of soil? Anyone want to take a guess?
More?
Somebody guess more? Anyone want to guess something else? That's a hint that more is not correct.
Less?
Less. How about 1 gram of human feces? More or less than 1 gram of soil?
More.
More, it's correct. On 1 human being, more or less?
Definitely more.
More, because obviously 1 human being is going to have more than 1 cubic centimeter. And then the entire planet Earth, of course, will have a lot more. So for ocean water, we have about 1 times 10 to the 6 microorganisms per 1 gram of ocean water. And that's less than 1 gram of soil. For feces, we have about 1 times 10 to the 11th microorganisms per gram of feces if the feces is very fresh. Okay, and it's actually 55% of the wet weight of the feces. So when you poop, most of that poop is actually microorganisms by weight. Okay. Now that differs on very dry feces, like if someone were to defecate in the desert and then it would dry out, you would only find about 1 times 10 to the 6 microorganisms. But when it's very fresh, it's very up to 1 times 10 to the 11th. On a human, you can find 3.8 times 10 to the 13 microorganisms on the human. And this will be largely on the surface of the human, as well as inside the human, like inside their gut, inside the mouth. On the entire planet Earth, we have 1 nonillion microorganisms. And I'm only talking about cellular microorganisms. A nonillion is 1 times 10 to the 30th, meaning 30 zeros following that 1. In comparison, on the planet Earth, this number is a little old. We have a little bit less than 8 billion people. So somewhere between 7 and 8 billion, closer to 8 billion now. Any question about any of that? All right, let's talk a little bit about microorganisms and disease. If a microorganism causes disease, we call it a pathogenic microbe or a microbe capable of causing disease. Any question about any of that? Now, you should realize that only a small percentage of microorganisms on the planet are capable of causing disease. And when we're talking about disease, it is important to distinguish between infectious diseases and inherited diseases. An infectious disease is one that is caused by a microorganism. What is an example of an infectious disease?
Tuberculosis?
Say again.
Tuberculosis?
Tuberculosis is an infectious disease. I should mention an infectious disease is one that somebody can get and then spread it to somebody else. Okay, that's an infectious disease. An inherited disease is a disease that you were born with, meaning your genes determine an inherited disease. What is an example of an inherited disease?
Huntington's.
Huntington's disease, or Huntington's cholera, is that what that's called? Is an inherited disease, meaning the child will be born with it. What's another example of a inherited disease?
Cystic fibrosis.
Cystic fibrosis is another inherited disease, meaning the child is born with that. Anyways, that's the difference between an infectious disease and an inherited disease. When we're talking about microorganisms, they are small. You can't see them with the unaided eye. How small are they? This slide is trying to show you a little bit. There's a bacteria, and it's a little bit less than 1 micrometer, also called 1 micron. This is something like E. coli, which is a little bit bigger than your average bacteria, although that's pretty close to the size of an average bacteria, which is only about 1 to 2 micrometers. They come in different shapes as well. Here's another microorganism, and this one is bigger. That's 50 micrometers, and this is bigger than 50 micrometers. In fact, I'd say that's bigger than 100 micrometers. There's another microorganism. This looks like it might be a red blood cell, or excuse me, a white blood cell. And this is even bigger. Well, by diameter, it'd be bigger. It might not be bigger by length. Let me see, this is a little bigger than 100. And if that's 10, yeah, the length here is less than 100, so that's longer. Here's another microorganism. This is a colonial microorganism. I'll blow that up a little bit. And each dot here is a cell. And so this is a colony, and this is the parent colony, and this parent colony has 1, 2, 3, 4, 5, 6 baby colonies inside it. This colony will open up to release the babies, which will then go out and colonize different areas to grow. This is Volvox, if you've ever heard of that. And then this is a T lymphocyte, CD4 lymphocyte. And then these little red things would be the HIV virus, which is also a microbe, but this would be a single cell, which is a microbe. All right, Microorganisms play numerous beneficial roles on the planet Earth, and we talked a little bit about these. They can decompose organic waste, break it down, and recycle the nutrients. Sorry about that. Microorganisms are producers in the ecosystem. That means they can make their own food. And these producers engage in either photosynthesis, where they convert light energy into the energy in sugar, or they can convert chemical energy inside a chemical, take that chemical apart and use it to make sugar. And these are the chemosynthetic microorganisms. Only bacteria and archaea are chemosynthetic, and we'll talk a little bit more about them later in this lesson. The point is that producers can make their own food given either light or a chemical. And then that allows life to continue, because like humans would then eat these producers, or another organism which eats the producer. Like if you eat a cow, the cow is eating the grass, which is a producer that engages in photosynthesis. Any question about any of that? All right, we mentioned that microorganisms are extremely important in the nitrogen cycle, and the recycling of nitrogen on the planet Earth largely happens because of microorganisms. Microorganisms can also be used to produce industrial chemicals, such as ethanol that you put in your car, or acetone. Microorganisms can be made to produce insecticides, or they do produce insecticides. We use microorganisms to produce food for us, such as fermented foods like vinegar, cheese, leavened bread, and beer. All of these happen because of microorganisms fermenting the food. Microorganisms are used to make products in manufacturing, like cellulase is generally made by microorganisms. And let's come down to here. You can use microorganisms to make other products that are used by humans, like in medicine, such as insulin. All human insulin made today, at least in the United States, is made in a microorganism. And what they did was they actually cloned out the human gene and put it into a microorganism, and then the microorganism makes the human insulin. And then they purify it and then give it to the patient. We can also use microorganisms to produce antibiotics. And we'll talk a little bit about that much later in the term. I'll mention that penicillin G is an antibiotic which is made by a microorganism. Any question about any of that? All right, didn't hear anything? So let's talk a little bit about biotechnology and genetic engineering. You should know that biotechnology is simply the use of microbes and other organisms to produce food and products for human use. So biotechnology is actually centuries old, where we use, I don't know, an organism to produce food for human, like kimchi and sauerkraut. That's centuries old, and that's using a microorganism to make the food. And that we can call biotechnology. Genetic engineering is a fairly new technique of biotechnology. And this is using microorganisms in the lab where we alter them to make products for us. Applications of genetic engineering include producing a variety of proteins from bacteria and fungi, such as producing vaccines, various enzymes, I don't know if you know of any enzymes which are made by bacteria, but you can sometimes buy the enzyme and see that it's made by a bacteria. Another example would be, oh, I had it. Oh, we use a fungi to make antibiotics. Like fungi makes penicillin G. That's the fungi is a microorganism. We can use genetic engineering to engage in gene therapy. A child is born with a defective gene, and we can insert that gene into the child and then correct the genetic deficiency. That is called gene therapy. And that has now been used for over 20 years. You can genetically modify bacteria, such as to protect crops from insects and from freezing. Like you can put an antifreeze in the bacteria and then spray the bacteria over the crop, and then the crop will be less likely to freeze. You need a colder temperature. Boy, what just happened? Can you guys hear me?
I can hear you.
All right, I just lost my screen for some reason. Not the sound, fortunately. All right. So let's talk a little bit about microbes and human welfare. We already mentioned that microbes help humans by making products for us and then recycling nitrogen on the planet Earth. Microbes also are important in recycling carbon, sulfur, phosphorus, and other nutrients on the planet Earth. So when a plant or a tree dies, those nutrients aren't locked into the dead trunk of the tree. The microorganisms will recycle those nutrients and then make them available to other organisms. Any question about any of that? The microbial ecology was first learned in the 1880s by Winogradsky and Bajorink, and they showed that microorganisms were important in recycling nutrients in the ecosystem. So they looked at nitrogen and found that it moves from one place to another in the earth because of microorganisms. And maybe we'll talk a little bit more about that later, where Winogradsky was the leader and Bajorink. What are those called? A column of mud. And you look at it, and it has a ecosystem of microbes in it, and they move nutrients from one form to another. So they were the first to show that microorganisms recycle nutrients in the ecosystem. Microorganisms are also important to human welfare in bioremediation. Bioremediation literally means to remedy using life. So life and to remedy. And that's like using living organisms to deal with an environmental problem. Like this pollutant, that's an oil spill, and they're spraying microbes on it to break down and digest the oil and then recycle it. meaning recycle the nutrients in oil by changing the oil into carbon dioxide and water. We've already mentioned that microbes break down sewage, recycling it. And then other pollutants can be either broken down and recycled or converted to a less toxic form. Like oil can be broken down and recycled. Mercury is an element, so it can't ever be recycled exactly, but it can be converted from one form to another. The microorganisms can make it less polluting, less toxic. If you don't know mercury is toxic to humans, the microorganisms can make it less toxic. Any question about that? If you don't know what toxic means, it means it's harmful. I mentioned that microbes can be used to make biological insecticides. These are alternatives to chemical pesticides, and generally they're less toxic or not toxic to humans. And you can use these biological insecticides to prevent insect damage to crops and prevent disease transmission, like among people. An example is Bacillus thuringiensis, shown here, a bacteria, and you just buy it in a product like that, and then put it on what you want to protect from many insects. Like my father bought Bacillus thuringiensis way back in the 1960s. And I guess that's dating my years because I can actually remember that, although I was very, very young. But I remember my father treated the apple trees with Bacillus thuringiensis to prevent the apples from getting worms. The trouble was he only treated the plants, the trees, one time, and you do need to use Bacillus thuringiensis about every 2 weeks if you want the apples to not have worms. And then on top of that, he was living in Western Oregon where it rains a lot. And every time it rains, it washes the Bacillus thuringiensis off the apples. So you need to treat the apples every 2 weeks and every time after it rains. And my father didn't do that. In fact, I think he only treated the apples once. And so they were wormy, but they were less wormy than if he never treated them. Okay. Any question about any of that? And then what's good about Bacillus thuringiensis, although it is fatal to many insects, it is harmless to humans and other animals, as well as plants. Any question about any of that? If not, let's talk a little bit about classifying of microorganisms. Here we're looking at a picture of the person who first started the classification system for all living things on the planet Earth. Does anybody know his name? I'm
His
sure
last
you
name
got
is
that
Linnaeus?
covered in Biology 160.
Linnaeus?
Yes, Carolus Linnaeus was his name, and he liked Latin so much that he actually Latinized his name. And I think it was something like Carl Linné, and he changed it to Carolus Linnaeus because he liked Latin. Anyways, he established the scientific nomenclature where each organism has 2 names: the genus name and the specific epithet. Each species has two names, and Homo sapien is the species name for humans, and Homo is the genus name. Sapiens is the specific epithet. You need both to be the species. This system is called the scientific nomenclature because we're still using it today to name things, meaning it was so good, we're still using it today. It's also called the binomial name nomenclature because every species has two names. Bi meaning two, nomial meaning names, two names. And you can also call it the scientific binomial nomenclature. But generally, I'll just call it the scientific nomenclature or the binomial nomenclature. Carolus Linnaeus, not only did he begin naming things, he also started classifying things, meaning all living life forms. And we'll talk a little bit later about classifying. At the moment, we're just talking about naming. Any question about any of that? All right, no questions, let's move on. When you're using a scientific name, it should be italicized if you're typing it, or if you're writing it, like on the blackboard, you then write it and then underline it to let you know that it is a scientific name, which is very hard to write italicized, so you underline it. Any question about that? When you spell out the name, the genus name is always capitalized with a capital letter, and the specific epithet is always lowercase. An example of that is Homo sapiens. All words or names are Latinized and they're used worldwide, meaning every place in the world, regardless of what the language is, a scientific name will have the same name that's Latinized, meaning Homo sapien is used everywhere on the planet Earth to mean human. The name can be descriptive or honor a scientist, like Homo sapien is descriptive. Homo means walks on two legs or something like that. Sapien means wise or wise man. Descriptive. E. coli is both descriptive and honoring a scientist. Let's see, Escherichia is honoring, I think it was Esther, who was an early microbiologist. And then coli is descriptive because we can find E. coli or Escherichia coli in the colon. Okay, Staphylococcus aureus is descriptive. It literally means golden aureus because the cells have a yellow color, and then coccus means ball or ball-like because they're in the shape of a ball. And then Staphylococcus means it grows like a grape because Staphylococci look like a cluster of grapes. They're little round grapes and they're stuck all together like a cluster of grapes. Any question about any of that? Now, after first use, the scientific name may be abbreviated with the first letter of the genus, a period, and then the specific epithet's name. So the first time you use Staphylococcus aureus, you have to spell it out. And then all other times you use it, you may say S. aureus. And then E. Escherichia coli, the first time you use it, you're supposed to say Escherichia coli. And then all other times you can abbreviate E. coli. Now, some words are so common, like E. coli, that even among scientists, the first time they use it, they will just say E. coli, because everybody knows what we mean when we say E. coli. And for some of you, you might not have known that E. coli stands for Escherichia coli. But all other cases, you should spell it out the first time and then abbreviate. Any question about any of that? All right, let's move on to the classifying of microorganisms. You can classify all microbes into different groups. And let's take a look at the classification of first all living organisms on the planet Earth. So when we start with all living life forms, the first classification we're going to discuss is the 3-domain system, where all living things can be broken down into one of 3 domains. The 3-domain system is sometimes called the tree of life because it branches like the branches on a tree. And yeah, this is a funny looking tree because it has 3 trunks, but I'm sure you've occasionally seen a tree with 3 trunks. The point is they all branch. The 3-domain system is based on genetic data, meaning we determine which domain a species belongs in by sequencing the DNA of the species and then checking the sequence. All of the bacteria within the domain bacteria have a similar sequence of DNA with other members of this family or domain. I should say domain. And this member belonging to the domain bacteria will have DNA sequence differing from the other domains, archaea and eukarya. So the domain is based on genetic data, the sequence of DNA. Any questions about any of that? And I guess the other thing is all living other life forms in this domain will have a similar sequence of DNA, much more similar to another member of this domain than to another domain. So when we're comparing the DNA sequence of humans to the DNA sequence of, let's talk about a slime mold, for example, or even an amoeba, the DNA sequence will be much more similar between the amoeba and the human than the DNA sequence between an amoeba and a bacteria, or an amoeba and an archaea. Any question about any of that? All right, if there's no questions, let's state we can then break down the domain into different groups. And generally, you break the domain into a kingdom. Now the bacteria, the domain bacteria has only one kingdom, which is domain bacteria. So in a sense, there is no break between the domain and the kingdom, because it's the same. And it's the same with archaea. But when we come to the domain eukarya, I forgot to mention there are 3 domains: bacteria, archaea, and eukarya. The domain eukarya can be broken into different kingdoms. We're going to use the 6-kingdom approach, which means 1 kingdom is bacteria, 1 kingdom is archaea, and these kingdoms are the same as the domain. And then the eukarya has 4, the domain eukarya has 4 kingdoms: plantae, animalia, fungi, and protista. Now I should state that the definition of a kingdom is not defined, and there is argument among scientists how many different kingdoms they are. We're using the 6-kingdom approach, but you could use a 5-kingdom approach or even a 3-kingdom approach. The 3-kingdom approach means there's the same number of kingdoms as domains, and then the domain and the kingdom are exactly the same. The 5-kingdom approach has these 4 kingdoms here, and then you lump these 2, bacteria and archaea, into the same kingdom. I'm going to suggest you don't do that because those are different domains, and the kingdom should be when you break down the domain. You can also have more than 4 kingdoms in eukarya. There's the 9-kingdom approach. So there's the 6, the 9, I think there's a 10, a 15, 19 or 20, something like that. Like I stated, there's not an agreement to how many kingdoms there are, or for that matter, what a kingdom is. Generally, when we're talking about the domain, there's not too much argument about what's a plant and what's an animal and what's a fungi, because these groups are monophyletic. But when we talk about a protista, all it is a eukarya that is not a plant, not an animal, and not a fungi. So the protista is a collection of other eukaryotes, meaning it's not monophyletic. And many of the higher kingdom approaches, what they do is they divide the protista into different kingdoms. And actually, here's one here. We can divide the eukarya into fungi, slime molds, whatever oma coats are, amoeba, chromis, animals, plants, ciliates, euglenozoa, plasmodia, microspora, and archaeozoa. Okay, you could divide that into each of these into a kingdom. And then there's higher orders of kingdoms where you start dividing plants into different groups, different kingdoms. All right, hopefully I haven't totally confused you. The point is, there is no definition for what a kingdom is. And then scientists argue about how many different kingdoms there are, as well as which kingdom a species belongs to. For example, we talked about the Oregon kelp or the Washington kelp. There's an argument among some scientists, is the kelp a plant or is it a protista or some other kingdom? Okay. Any question about any of that? All right. When we're talking about the 3-domain system, it's important to realize that there are 3 domains, but there's only 2 cell types. And cell type is the term that your textbook author uses. So when we have the domain bacteria, the cell type is prokaryotes. When we have the archaea, the cell type is prokaryotes. When we have the domain eukarya, the cell type is eukaryotes. Any question about any of that? All right. No questions. So let me state that your textbook author calls this a cell type, but other authors will call prokaryote versus eukaryote a super kingdom, something higher than a domain, where you divide all living life forms into eukaryote and into a prokaryote. And then you divide the prokaryotes into 2 domains, the bacteria and the archaea. I'm just telling you that because different authors treat it differently. That is not the way your textbook author treats it. He uses the 3 domain system and says that there are 2 cell types. And we'll talk a little bit later about prokaryotes versus eukaryotes. You already know, I think you already know, that a eukaryote has a nucleus and a prokaryote does not. All right, let's talk a little bit more about the domain bacteria. The cell type is prokaryotes. Bacteria, all of them have peptidoglycan in their cell wall. So if bacteria has a cell wall, you can find peptidoglycan in it. Any questions about any of that? All right. The bacteria mostly divide by binary fission. That's where the cell literally will divide straight into 2. For energy, bacteria can use an organic chemical compound such as glucose, and that's what we do. But bacteria, some of them can use an inorganic chemical like iron or ammonia or sulfate. And then some of the bacteria can get for energy, they can use photosynthesis. Most of the bacteria are either free-living decomposers or primary producers, meaning they make their own food, primary producers, or they decompose something. But some of the bacteria are parasitic, and some of them can cause human disease, in which case we call them pathogenic. Any question about any of that?
I have a question.
Go on.
Is binary fission, is that the same as mitosis?
Can you say that again?
Binary fission, is that the same as mitosis?
No. Mitosis is only seen in eukaryotes. Eukaryotes do not divide by binary fission. They divide by mitosis. Binary fission is where, we'll talk about it later, but the DNA will replicate and then one molecule will move to one side of the cell. The other molecule will move to the other side of the cell. And then the cell will literally start dividing in 2, splitting in 2. And then one cell will become 2 daughter cells.
Okay, thank you.
Okay. All right. If there's no further questions, let's talk a little bit about the domain archaea, also called the archaebacteria. They are all prokaryotic, meaning their cell type is prokaryotes. Does anyone know, do the archaea have peptidoglycan in their cell walls? Anybody know?
No?
You are correct. The archaea do not have peptidoglycan in their cell wall. Some of the archaea are found in extreme environments, like the methanogens that produce methane, the extreme halophiles, which live in very salty conditions, like the Dead Sea between Jordan and Palestine. Some of them are extreme thermophiles, where they live in very extreme high temperatures. By a thermophile, we're talking about over 95 degrees Celsius. Remember, 100 degrees Celsius is the temperature water boils at. However, when we look closer, we can find the archaea in all of the Earth's biomes. We can find them everywhere on the planet Earth, and they do make up a significant proportion of the microbes in ocean water. So they're a significant portion of the plankton, and they are also a significant portion of the microorganisms in the soil. Any question about any of that? All right. Does anybody know, are any of the archaea pathogenic?
I'm gonna guess no.
That is correct. None of the archaea are pathogens. We don't know of any examples of an archaea which is pathogenic. For this reason, other than this lesson, we will not be talking much about the archaea. It's not that they aren't important, it's just that the focus of this class is to discuss pathogenic microorganisms, because many of you are going to go working in the health professions. And so the focus of this class will be talking about the bacteria, because some of the bacteria are pathogenic and none of the archaea are pathogenic. Any question about any of that? All right, if there's no further questions, let me state that we need to talk about the eukarya. We already mentioned that the eukarya can be divided into 4 kingdoms: the plants, the animals, the fungi, and the protista. And this is just the number of kingdoms I'm going to work with. It's actually what biologists used before they started arguing about how many different kingdoms there were. So we'll talk about the 6 kingdom approach, and 4 of them belong to the domain eukaryotes. All of the eukaryotes lack peptidoglycan. So even if the cell has a cell wall, it does not have peptidoglycan. Peptidoglycan is only found in domain bacteria. Any question about any of that? We'll talk a little bit about each kingdom. First, we're going to talk about the fungi. You don't need to know about chitin. The fungi use an organic chemical for their energy, such as glucose, which is similar to what we do. You can have a multicellular organism like this mold here, or a mushroom, multicellular, or you can have a fungi which is unicellular, like yeast, shown here. Of the fungi, most of them are decomposers, but some of them are pathogens. Of the kingdom Protista,
I wanted to let you know it's 6:15, Dr. Volp. I didn't mean to interrupt, sorry.
Yeah, I want to finish this slide and then we'll finish. Of the kingdom Protista, there are plant-like protists, animal-like protists called protozoa, and then fungus-like protists. The plant-like protists engage in photosynthesis for obtaining their energy needs. They generally produce molecular oxygen as well as an organic compound like glucose. And they have cellulose in their cell walls. Cellulose is what you find in the cell walls of a plant. The protozoa include the amoeba, the paramecia, the euglena. They absorb or ingest organic chemicals, such as glucose. They may be motile via pseudopods, shown here, cilia, which actually this one has it, although you can't see it, or flagella. Some of the protozoa are parasites, causing disease in humans. An example would be amoeba, causing amoebic dysentery, or malaria, another parasite. We're not really going to talk about the fungus-like protists, but there are some fungus-like protists. I can actually mention one, and that is the water molds, which caused the Irish potato blight, which caused famine in Ireland and a whole bunch of Irish to migrate to America. That's an example of fungus-like protists. All right, any question about any of this? If not, I'm going to stop here and we will continue with this slide next time. And I'll see you in the lab at 6:30. All right,
Thank you.
goodbye.